Last fall I did a video about SSRI antidepressants and the desensitization that is seen with 5-HT1A autoreceptors. I was really interested in that topic, and since then I've been doing more research. And so the topic that I was next interested in, naturally almost, was the serotonin transporter, SERT. And serotonin is also called 5-HT. That's kind of the abbreviation for serotonin. So 5-HTT is the serotonin transporter, or SERT. And what it does is, in the name, it transports serotonin. And so it's the serotonin transporter. Right up here I have a picture of serotonin, the molecule. And that's these blue dots down here and the synapse area between the presynaptic and the postsynaptic neuron. And so these are the receptors on the postsynaptic side. And you would see the presynaptic autoreceptors over here, which I talked about in my last video. But you also on the presynaptic side are seeing SERT, or the transporter that's taking serotonin from the synaptic area and putting it back into the presynaptic area. And so the idea with SSRI antidepressants, which I'll touch on a bit later, is their ability to block this action right here, leaving more serotonin in the synaptic area. And this is all happening in the midbrain. So this is a picture of a rat's brain, and this is all centered around the dorsal root nuclei and projections that are directly from that. And so that's why you're seeing this happening in serotonergic cells that have serotonin. And the expression of SERT is implicated in SSRI treatment pharmacology. As I talked about a bit on the last slide, SSRI antidepressants are targeting SERT, and so they're blocking SERT. They're blocking its ability to transport serotonin from the synapse back into the presynaptic membrane. And by doing so, they're increasing the amount of synaptic serotonin. What's a little bit confusing is that you also see around the 10% decreased level of the SERT expression in patients with major depressive disorder, or MDD. And the severity of depression is associated with the amount of SERT reduction in the amygdala. So this is another way to view it in a comical way. So right here is the transporter area, or SERT, and this is kind of giving a channel for these sheep to cross through from the synaptic area, which is right here, into the presynaptic area, which is up here. And so what the medication is doing is blocking this ability to transport. So it's allowing the sheep to build up on the synaptic cleft land in this picture, which would be right here between these two neurons. So this picture shows the binding of SERT before and after the treatment with the SSRI citalopram. Areas that are red here are areas that have higher SERT binding. And so you can see after this 4-week period, it's decreased. And so that's showing less SERT activity after antidepressant treatment. And you can actually see that again here in a different study where they looked at the SERT density as a function of time. And they also looked at the SERT mRNA levels, or how much SERT is being expressed genetically. This is all, these first 20 days here are all days of treatment. And then these last 20 days here, from 20 to 40, are days of washout. And one of the easiest ways to check how one person can respond to differing levels is by recent studies that have looked at male to female and female to male transsexual sex reassignments. Split that up into two hormones in particular, and that's testosterone and estradiol. During a female to male transsexual sex reassignment, testosterone treatment is usually the main way to get that started hormonally. After looking at 1 and 4 months after the beginning of that treatment, there was an increase in SERT binding and the amygdala, caudate, putamen, and medial Rolf nucleus. And then flipping over to estradiol, which is abbreviated here as E2. What happens a lot in these male to female reassignments is they take an estrogen, which is estradiol. It's related to estrogen, and then an anti-androgen, which is blocking androgens in the brain. And so after 4 months of this, they noticed decreases in SERT binding in the insula and mid-cingulate cortex. This wasn't mediated by estradiol because increased E2 levels correlated negatively with decreases in regional SERT binding. So that's saying that if you had high levels of that E2 being administered with the anti-androgen, you're having decreases in the amount of SERT binding that is lost. So that indicates a protective effect of the estradiol, or the E2, against SERT loss. A subsequent study looked back and said, so if you have these ovarectomized, or mice that have their ovaries removed, these female mice without ovaries, what would you have for SERT density in those mice? And they have reduced SERT density. And they also, because they have their ovaries removed, they have low estrogen and estradiol levels. When you increase E2 or estradiol, you see that SERT density is increasing. And so that's showing you that the increases in SERT are mediated by estrogen receptors. And so the question you might ask is, well, then what are you seeing here in testosterone? How is testosterone working through an estrogen receptor? That doesn't make sense. Estrogen is seen as typically, and this isn't really correct, but as just a female hormone, and testosterone is seen, again, incorrectly, as just a male hormone. They're actually in both sexes. As you can see in this diagram here, these are the three brain regions that I talked about in the last slide: the amygdala, the caudate, and the putamen. And this is for female to male transsexuals, so this is when they have just testosterone treatment. And you can see here a significant increase in the SERT binding potential, or basically how available it is to bind serotonin. And you can see here after 4 weeks and after 4 months, this increase in all three regions. One thing that we've noticed is that DHT, the androgen DHT, doesn't increase SERT expression. So then why is testosterone increasing SERT expression? And how could this be tied into the way that estradiol is protecting against SERT loss in female animals without ovaries? And so we have to look down here at this steroid conversion pathway. And so when you have testosterone here, it can either be converted with 5-alpha reductase into DHT, or dihydrotestosterone, or aromatase can aromatize it into estradiol. This estradiol that's been given in the male to female transitions is very close to the testosterone that's being given in the female to male ones. And this aromatase enzyme here is all that's separating testosterone from estradiol. And because, as I just said, DHT doesn't increase SERT expression, but testosterone does, and estradiol does, it seems that testosterone being aromatized into estradiol is what's actually causing the increase in SERT density and messenger RNA. It suggests that the SERT increase is therefore mediated by estradiol, or E2, and that's through estrogen receptor activation. In these hormone pathways, aromatase catalyzes testosterone being aromatized to estradiol, or E2. And so the SSRI fluoxetine inhibits aromatase activity in multiple assays. SSRIs tested inhibited the activity of aromatase, and this might explain the lowered E2 levels. So because estradiol is able to protect from SERT loss, it could be plausible that one of the ways that SSRIs are decreasing SERT levels is through the inhibition of aromatase and the decrease in estradiol availability in the brain. And these are the sources that I used for this presentation. Thank you for watching. If you want to check out more on my channel, you can subscribe, or if you want to look back at my older videos, I'll put them up here as well.